The chef doesn't cook with whole, unpeeled vegetables. Prep cook washes, peels, chops, and measures before anything hits the pan. Machine learning models are the chef. Feature engineering is the prep cook. Raw data, gene counts spanning 5 orders of magnitude, categorical variables mixed with continuous, skewed distributions next to normal ones, need preparation before any model can use it effectively. Raw gene expression data has gene A with counts ranging from 0 to 50,000 and gene B ranging from 0 to 5. If you run KNN or PCA on raw counts, gene A dominates. Its large values create enormous distances, making gene B invisible. Categorical variables, like treatment group, can't be multiplied by a coefficient without encoding. Skewed distributions violate the assumptions of many statistical tests. Preprocessing transforms the raw data into a format where every feature contributes fairly and algorithms work correctly. Gene expression counts are right skewed, most values are low, a few are very high. The log transformation, typically log 2 of x plus 1 to handle zeros, compresses the right tail and spreads the low values, producing a more symmetric distribution. This is why most single-cell RNA-seq workflows start with log normalization. SCTransform takes this further. It fits a regularized negative binomial regression per gene, modeling the mean variance relationship and producing Pearson residuals as the normalized values. SCTransform is now the recommended normalization for SCRT because it accounts for the count nature of the data better than simple log normalization. Z-score standardization. Subtract the mean and divide by the standard deviation. Every feature now has mean 0 and standard deviation 1. This is critical for distance-based methods PCA, KNN, SVM, and K-means all use distances between observations. Without standardization, high-variance features dominate the distances. Gene A with variance 10,000 rounds out gene B with variance 1. After z-scoring, both contribute equally. In your pipeline, SCRT scale data performs z-scoring before PCA. Min-max scaling squeezes values to the 0 to 1 range, subtract the minimum, divide by the range. It preserves 0 entries, a 0 stay 0, which matters for sparse single-cell RNA-seq data where most entries are 0. The downside, a single outlier stretches the scale, compressing all other values into a narrow band. Min-max is useful when you need bounded inputs, such as neural networks with sigmoid activation functions that output values between 0 and 1. Robust scaling uses the median instead of the mean and the interquartile range instead of the standard deviation. Few extreme outliers don't affect the median or IQR, so the scaling is stable. For clinical data with extreme lab values, a creatinine of 50 when most patients are around 1, robust scaling prevents the outlier from distorting the scale for everyone else. It's the default recommendation when you suspect outliers but don't want to remove them. Log transformation for skewed counts. Z-score for uniform scale. Min-max for bounded range. Robust scaling for outlier resistance. SCTransform for single-cell RNA-seq counts. Each transformation prepares the data for a specific algorithmic need. Next, encoding categories and selecting features.